Other people are just crashing, and so it just doesn't really work so well. But hopefully this next, so we have a practical next week. So often don't be confused. If at any point during this lecture I say next week, and I refer to something you're going to be learning next week, I'm just misspeaking. I mean in 2 weeks. Because next week to me is renal physiology, but it's really 2 weeks, right? So we got a practical next week. You're not learning anything new next week. You're just taking an exam and that's it. We'll talk about the specifics of that exam when I get done with this lecture. But this week, all anatomy, that's what's going to be on your practical and for the renal system. Not too bad, about 3 pages of stuff that you probably get introduced to in lecture. We're going to try to overlap a little bit of function because I'm trying to lay the groundwork this week for what we will be learning in 2 weeks, which is urinalysis. So we're going to take the basic anatomy and a little bit of physiology of the kidney, really, and try to figure out what it means when stuff shows up in your urine and try to say, okay, here's what's showing up in my urine. What does that mean specifically in terms of how my kidneys are functioning? Because urinalysis is a really cool thing. You don't have to put an IV in, you don't have to draw blood, you just pee. And so it's just very convenient. And so it's a very, very useful way to get an idea of what's going on. And since your urine originates from the kidney, it's really important to understand how the kidney works. So let's talk about the kidney in general first. So we'll go to just a picture that I'm sure you guys have seen before at some point, either this week or other times in your life, where you're looking at just where the kidneys are. So kidneys are not particularly symmetrical. They're usually offset a little bit. One of them's higher than the other. It's not always the case. This is what the case is in children, but then as we age, we tend to start laying down peritoneal fat that can push our organs around just a little bit. But for the most part, this is the situation you're looking at, mirror image organs. Some people have a third kidney. That's a fairly common variation where you would actually have an extra kidney. Then some people have a U-shaped kidney. It's called a horseshoe kidney. That's actually what you'll read in patients' notes, horseshoe kidney. It actually connects across the spinal column. So it's anterior to the body of the vertebrae, and it actually forms a horseshoe shape. But for and then some people are missing a kidney from birth, and then some people have a kidney transplant, where often they will not actually remove one of the kidneys. They'll leave it in place, but they'll tie in the donor kidney on one side or the other. And the third kidney, the newer one that you've received, could either be placed lower in the abdominal cavity, or they push the old one down a little bit and place the other one in. It kind of just depends on the individual and the strategy of the surgery. So there's quite a bit of potential variation that you may see in kidneys. However, what's interesting is that even with all those variations, you don't see a huge difference in function. So someone born with a single kidney or born with 3 kidneys doesn't actually show a significant difference in function. So there's some kind of self-regulating mechanism that our body has that even with less kidney to do the work, we can, for the most part, get the job done up until we get to the point of so much reduction in functional kidney mass that we go into renal failure. We'll talk about why that happens when we get to the nephron, but it's kind of interesting that way. There's a lot of anatomical variation, but less variation than you would expect in terms of function. The big key thing with the kidneys is that they are somewhat well protected by the 11th and 12th rib, right? Our floating ribs cover the lower portion of them. 9 and 10 will, in some people, actually protect the top part where the adrenal glands are up here. So kidneys can be injured due to trauma. Bicycling accidents are notorious for them for both spleen and kidney injuries because the handlebar will go right up under the ribs, and then you usually fall forward on a bike rather than backwards, and so you'll just get stabbed basically with your handlebar. When I used to ride BMX, I had a friend that went straight through his stomach. He didn't have bar ends on, and so it just went. It didn't go in that far. It went in like 1.5 inches, but just straight like a cookie cutter right through his skin, and it was not good for him. But luckily, he didn't touch any of these large arteries or veins, because if you rupture a renal vein or artery, normally you're more worried about arteries because the pressure is so much higher, but even the vein in this case would be so traumatic. You would be losing blood at a rate that you may not be able to do anything about that before you would die. And so the kidneys, in some people, actually drop down. It's called ptosis. You're gonna have to stand, or sit. You can sit on the back table if you want. So ptosis is a term that we learned when we briefly mentioned with the eyes. It's a drooping eye. And so ptosis is a Greek term that means to lower or something like that. And so the kidneys can also actually come lower than they're supposed to be. This can be damaging because it exposes them even more to trauma. And this happens from normal, like laxity of the support of the organ, but it can also happen from extremely low body fat. And so long distance runners can run into this case where they get so low in body fat that they do not have this what's perirenal adipose, which is part of the capsule that surrounds the kidney. They get so little of it that the kidneys sort of become loose and are allowed to drop down more. For runners, it's not a huge issue because it's not a contact sport, but there are some boxers that run into this issue. Somebody like Floyd Mayweather, I'm not saying that he does, but somebody, if you know who that is, he's a smaller guy, very low body fat percentage, but in a sport where punching your kidneys is a strategy. And so for people that have this issue, quarterbacks too in the NFL, they can either have them surgically elevated again or you discontinue contact sports because of the likelihood that at some point something's going to hit the kidney enough to cause it to bleed, and if the kidney bleeds, it bleeds a lot. And it doesn't tend to repair due to trauma very well at all. So that's kind of the big picture. These organs are retroperitoneal. Let me pull up another picture that will better illustrate what that means. So in your abdominal cavity, which is what we're looking at now, there are two major subdivisions, the peritoneal cavity and the retroperitoneal cavity. So if we look from above, and cut at a totally different angle now, surrounded in kind of a light blue, is your peritoneal cavity. That's where all of your small intestine is, all of your stomach, your liver, part of your large intestine, the transverse colon actually leaves. That's what these little things are, is actually the transverse colon leaving the peritoneal cavity. But for the most part, it's where your digestive organs are. It's like a big garbage bag full of digestive organs. And then behind that garbage bag, though, there's a whole space full of other organs like your adrenal gland, your kidneys, and part of the colon. And so those things that are behind this blue line here are referred to as retroperitoneal. And so that's just something to know, and it is relevant because this is one reason that these kidneys have more exposure to trauma. It's usually right in here where you could best damage a kidney because they're kind of protected from the back by musculature usually. But also it protects the kidneys to some extent from infection because if you remember all the bacteria that are actually in the peritoneal cavity, really a trash bag is a good analogy because there's a lot of bacteria, both good and bad, that are in the digestive system. And so if you have a bacterial infection in the peritoneal cavity, then it will tend to remain isolated there. Whereas, which is a good thing because a kidney infection is a really, really bad thing. Now, if you do have an infection somewhere in retroperitoneum, then it's going to very easily travel to the kidney, but it creates just a subdivision, two different separate areas. And then surgically, it actually is a really good thing because you don't have to breach the peritoneal cavity in order to do a kidney transplant, which means you basically leave the digestive organs intact, and so it actually is very convenient for access to the kidney itself because it is, again, retroperitoneal, just behind the peritoneal cavity. So let's see, on the website today I have 3 different videos that go over each one of the main models that we're going to be looking at. I'm going to use this to walk through some of the general structures of the entire kidney. So we'll start with this image right here, which is identical to the model that's back there next to Dan. So what this is not showing, remember around the kidney, it's not so clean and nice. You actually have this capsule, which is in dark brown here. It's just like the fibrous capsule that's around a kidney bean, right? It's really similar to that. And one thing to notice about the capsule itself is, yes, it's made of perirenal adipose. There's a fat component to it. But the fibrous component, the part that's really like the fibrous component around a kidney bean, is not stretchy at all. And so you have a lot of blood coming through the kidneys. So you have 20% of every heartbeat at rest. 20% of the volume of every heartbeat goes to the kidneys. So this is a big artery full of a tremendous amount of pressure. So there hasn't much, remember that graph I showed you how pressure starts out really high in the big arteries and then it drops rapidly as it goes through the arterioles? There hasn't been much time for blood pressure to drop by the time it gets right here to the renal artery. And so if you have variations in your blood pressure or a blood pressure that's chronically high, any large blood pressure or change of volume to the kidney can't be accommodated or can't be dealt with by stretchiness, right? So you breathe in a little bit too much air, great. Your lungs can accommodate that. You send a little bit too much blood to the heart, big deal. Just the Starling effect happens. You stretch the heart, it pumps harder. So there are a lot of organs where sending too much volume or pressure, organ can deal with it because it's stretchy. Your kidneys can't. And so this is why high blood pressure, we think of like that being an issue with the heart because the heart might have to work too hard. But that's sometimes the case in the elderly, but for people that have genetically kind of higher blood pressures, the things you worry about instead are the fact that the kidney works to regulate your blood pressure, but it's not perfect. It sometimes makes mistakes. And when it makes mistakes, it gets a little bit more pressure than it was expecting, and you damage a little bit of the kidney. Well, people with higher blood pressure pay a higher price for making those little mistakes. So that's the big difference between males and females, because females have a significantly lower blood pressure. And so our male and female kidneys are making the same number of mistakes, but people with higher blood pressure pay a higher price for those mistakes. And so that's the inelasticity of the capsule is why everybody pays a little bit of a price. If the kidney was a little bit more stretchy, that may not be the case. You also have a posterior renal fascia that's not illustrated here, but that's what anchors the kidneys to the abdominal wall. This is what can contribute to ptosis, because if it were too long or too lax, it can actually allow the kidney to descend into what's called ptosis, renal ptosis specifically. Now there's a big list, there's a bullet point there with a bunch of lists of arteries and veins. I'm going to push that off till next week. We're going to talk about the specific blood flow next week, so you can cross it off for this week, but we're going to come back to it. And again, I don't mean next week, I mean in 2 weeks. In 2 weeks, we will come back to the specific blood flow as a way to sort of review what we talk about today, because in 2 weeks, that's a long time from today, and that's the next time we're going to be talking about the kidney. All right, so when we look at the macroscopic kidney here, you've got another hilum, just like we saw in the lungs, with the blood vessels. And in this case, we have a third major vessel that would be carrying urine. This is the ureter, indicated by number 3. So those are all entering and exiting via the hilum, the little dent in the organ that's kind of the doorway for all the major vessels. And there's also a tremendous amount of nerves and lymphatic vessels that are going through there, but you can't see them in this model because they didn't illustrate them. Related to the hilus. So hilus is like the doorway right here. You have two terms that are extremely easy to confuse, and it is the renal pelvis and the renal sinus. And it's a tough one, so let's go over it and spend a little bit of time on it. So you have renal sinus at the bottom of page 15, and then later on in your packet you have renal pelvis. So let's talk about the difference between those things. So renal sinus, I want to explain that one first because I feel like that's kind of the stage that we're at here. So if I were to grab this artery, renal vein, and the ureter, just grab it and rip it out of the organ and leave everything else intact. Just rip those out, all the red, all the blue, and all of this sort of tan colored stuff. Just get rid of it, but leave everything else intact. There would be a space left behind, and that's the renal sinus. So the renal sinus is actually occupied by your blood vessels. There's fat in there too, and also this big whitish tan structure in here. The 3 is the ureter, and 4 is actually what's called the renal pelvis. It's this big common area that leads to the ureter. So the confusing part is renal pelvis and sinus. Sinus is like a potential space. It's a space that doesn't actually exist anatomically, really, but it can be used, and you may see it written, because if someone has like a benign tumor or something, people sometimes have growths and tumors in their kidneys that you'll have imaged. You'll see a radiologist write, maybe if there's a benign growth here that was just slowly growing, they would say it maybe protrudes 3 centimeters into the renal sinus. It wouldn't necessarily protrude into the renal pelvis, because this pelvis area, number 4 is indicating the renal pelvis. It's a closed space that is actually where all the urine is being drained into before it goes into the ureter. So the renal pelvis is in the renal sinus. They're not exactly the same thing. And so it's kind of a weird one because renal sinus is a potential space, but you will see it, potentially. you'll see it in the future. Does that make some amount of sense?
So,
All right. Yeah.
does the pelvis only contain the ureter type tissue, or is it
Correct.
that whole?
It's just like a continuation of the ureter. I mean, it's going to flow this direction, but it's like an expansion of the ureter that's collecting urine from all the subcomponents of the kidney.
Right.
So yeah, tissue-wise, it looked the exact
Whereas the
same.
sinus is like the whole cavity, where a cavity would be if you took all of it.
Exactly. It's where cavity would be if you ripped everything out. All right, so I'm going to skip up to page 16 for gross anatomy of the pig or sheep kidney. We're not going to do the dissection today. We're going to save that for 2 weeks also. But I do want you to still know these terms. So I'm going to hit each one of these terms right now, but from this model perspective. So we've already talked about the first few terms there. So renal artery, renal vein. Right there, again, just appreciate how much of your blood flow goes to the kidneys. That's why, I mean, if you imagine the blood flow, remember coming out of the heart, you go through the aortic arch and then the descending aorta. The first big exit off of the descending aorta, the really big one, I mean, it's as big as my thumb, each one of them is the renal artery. So you have a tremendous amount of blood pressure going to the kidneys and a tremendous amount of volume. So again, I also want to emphasize that your kidneys, at rest, it's 20% of your blood flow. When your cardiac output increases, like during exercise or anything like that, it will not continue to be 20%. The total volume going to the kidneys is supposed to be the same at all times. So as you increase your cardiac output, you get all excited and start sweating, you're not going to throw more blood at the kidney, you keep it the same. You're trying to, the goal, just like the brain, is basically to always send the same amount of blood, but during extreme physical activity, you do start to cut off blood flow to the kidneys. And so in extreme stressful situations that are prolonged, you can actually run into issues with that. But for the most part, the idea is always send the same amount of blood to the kidneys. All right, let's divide this organ into medulla and cortex. So we've heard these terms before. Cortex is always on the outside, medulla is always deeper. All you really need to know is how do we divide this? Well, if you look through here, there's these sort of pyramidal shaped structures. There's 1, 2, 3, 4, 5, 6, 7 of them. Those are called the renal pyramids, also called the medullary pyramids. And they are within the medulla. The reason I point them out for picking out the medulla is they're the easiest thing for your eye to go to find the medulla. So the medulla is the region that contains all of these medullary pyramids. So the pyramids are not the only thing in the medulla. There's other stuff in here, but it's the easiest way for your eye to pick it out. So always look for those medullary pyramids, and that'll tell you the division point, because at the top of these pyramids, I guess sort of at the bottom if we're considering it a pyramid, but at the outside, anything beyond that is going to be your cortex, indicated by B right here, going around the whole outside. So that's the basic division of the kidney. And functionally, it's an important division because there's very different things going on in the cortex versus the medulla. We'll talk about those when we get to the nephron.
Is there always 7 pyramids? Or
No, this is just, that's a good approximation from any kidney I've seen, but remember, this is just one slice. So they go off in all different directions. There's a nice model in the back that shows it more in 3 dimensions, because these pyramids are not like pyramids of Egypt pyramids. They're more like a makeup brush, like something you put blush on with. They're almost exactly what a makeup brush looks like. That's really what they have. They're domed at the end, and they go off in each direction, radiating in all directions basically, and they converge. So like the handle of your makeup brush would be connected at what's called the renal papilla. And so that's another feature associated with the pyramid. And it's basically just the little tip there and there. Really easily, it's indicated by number 6 in this model, there and there. So at the end of each one of these renal pyramids, there is a papilla. And that's a functionally really important thing, because these renal pyramids are constantly producing urine. And the papilla is just sort of dripping, drop by drop, all the urine that you're producing. It contains waste products that you want to get out of your body. So they're all coming off of the papilla right there and there and there and there. So that's where all of those nephrons of the kidney converge within the pyramid, is at the papilla. Now, the papilla itself, though, it doesn't just drain it into the retroperitoneal space. That the filtrate or urine it's technically not urine until it gets in the ureter, but it's basically the same thing. You might see it say filtrate, though, in your book. So properly, it would be filtrate, but basically urine, is drained into these little cups. There are these things that are over the papilla, and they're capturing that urine that's being formed. These are called minor calyces. There's one there, another one there, there. Every papilla has its own minor calyx. Calyx is singular, calyces is plural. So those minor calyces capture the filtrate. They merge together to a major calyx. That's what this one is because it's after two or more calyces have merged together. So that's a major renal calyx. And then all the major calyces converge together into the renal pelvis, number 4 in the picture.
Can you show the minor and major again?
So minor is actually kind of suctioned onto the papilla. Minor, 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 minor. And then when two of those minors merge together, like I think it'd be best seen right here, these two come together, this right here would be a major calyx, right at that point. And then all the major calyces merge together into the renal pelvis, number 4.
Yeah, Greg, did you say a minor would be 5B?
5B would that would be a minor right there. So those minor to major to pelvis, the renal pelvis drains into the ureter. And then from that point on, ureter goes to the bladder, then bladder goes to the urethra, and then you're into the toilet. Yeah.
So you said 2 minor will converge into a major?
2 or
always?
more. It
Okay.
could be 3.
Okay.
Yeah, it could be 3 all
So
coming
it's not
together.
like a
I'm sure it's
set.
really variable, yeah. Just know that once a minor, know that it kind of as a pathway. It's kind of like giving directions. Like this is the roadway that begins, you're starting from the renal pyramid, also known as the medullary pyramid, which drains into the minor calyx, then a major calyx, then a pelvis, then a ureter.
It's like a cul-de-sac.
It is kind of like a cul-de-sac. And then the other thing I haven't mentioned yet, it's not related to the blood flow, but it's another feature in the medulla, are the renal columns. And they are between the pyramid. That's what you should have in your notes. They're in the medulla only, but they're between the renal pyramids. And the renal pyramids are very, very delicate. They need to be oriented. As we'll see, this is where all the tubes, they really actually need to be lined up physically, so their 3-dimensional structure matters. And so they are kind of supported on the outside by the more connective tissue, is much more, less functionally important, but more structurally important renal columns. So these are kind of like pillars of strength in the organ that connect the deep regions of the organ near the renal sinus to the cortex and allow this whole organ to keep its 3-dimensional shape. Because if the kidney doesn't keep its 3-dimensional shape, it cannot function. And that's because what the kidney does is really based on mostly physical chemistry and osmosis. So its structure is really important to its function, and I'll compare that to the liver, or contrast that to the liver, where the 3-dimensional structure doesn't matter as much. Sure, the blood still needs to come in and go out, but the chemistry that goes on in the liver will happen no matter the shape or really the size of the liver. That's not the case for the kidneys. That's why trauma to the liver, though the bleeding could kill you, you're still going to be okay. Trauma to the kidney, not as much. All right, let's get a little bit closer. So now this is also a model that we have. We're zoomed in a little bit closer. Now we can begin to see the 2 different kind of nephrons. So you guys talk about nephrons. They're the functional unit of the kidney, and they are the most important part of this entire renal discussion. Because the kidney, unless you're going to be a surgeon or work in a surgical setting, the anatomy is good for a context, but you're probably not going to use it that much. What you will use constantly, regardless of profession, is the understanding of what a nephron is and what it does. Because the kidneys come up all the time. All the time. And they're going to have drugs or procedures or things, activities, anything that are going to affect the function of the nephron. And so we're almost to the zoomed in version of the nephron. That's kind of like the grand finale of this discussion. But we're just beginning to see the nephrons now. And it's good to see because this is where you see the cortex up here and the medulla down here. You can see that the nephron itself, right, it's got a glomerulus. It's basically a long tube connected to the glomerulus. We'll go through the individual components, but just notice the fact that the importance of the medulla is that it's the only place that contains the loop of Henly. So this loop of Henly, a long one here in what's called the juxtamedullary nephron, they're the bigger, arguably more important nephrons. And then this is what's called a cortical nephron because it's closer to the cortex. And it also has a loop of Henly, but it's much shorter. So what you should appreciate from this is that the loop of Henly, its main goal is going to be to create a concentration gradient to make a really salty environment within the kidney. And we'll talk about why we do that in a moment, but that's the main goal. This whole medullary area is actually very salty, and it's the loops of Henly that do that. And so it's cool to appreciate, I think it helps it kind of make a little bit more sense to appreciate that the only part of the nephron that goes down into the medulla is the loop of Henly. Now, 80% of your nephrons look like the one on the left, cortical nephrons. About 20% of them look like the one on the right. And the one on the right is more important for dehydration tolerance and things like that, things that have to do with water availability, mostly dehydration stuff. The one that you're going to learn more about is going to be the one on the right, because it's the more specialized one. And that's only for humans. We're at this 80-20 split. A lot of other animals that deal with, like living in deserts, you're going to see animals have a lot more of those, a lot more of the larger ones. All right, and then again, the blood flow is in this model, but we're not going to deal with that until later on in 2 weeks. All right, let's take a look at the nephron. So I'll show you a more realistic picture of the nephron first, but we're going to come back to this image because it's like too messy to actually have a more realistic view like this. So this is more what those two nephrons are really going to look like. We're going to look at it and talk about it in a second in a much more simplified way, but we're not leaving out any details. We're just being less honest about how complicated the structure is. Because for me to point out, like, where's the proximal and distal convoluted tube is kind of hard when you're looking at it in reality. But realize this is really what we're dealing with. A bunch of wadded up tubes with all sorts of blood flow around it. But we're going to take this and break it down into a little bit more easy to digest structure. And I encourage you guys to get your own drawing together for looking at the nephron. Something that looks, the reason I pull a picture like this is kind of the thing I would draw out when I'm trying to kind of understand and remember the different pieces of the nephron. It's not that it's realistic, it's just that it's simple and doodleable and will really help you guys when you're trying to organize your thoughts for both exams or when someone says, oh, this affects the proximal convoluted tubule's ability to do this. Wait, where was that again? You could sketch it out real quick just to give the memory to come back to you. All right, so just dwell on that for a second. I'm going to try and connect it to the stuff that we've already talked about because the kidney can be a little overwhelming at first. And specifically what the nephron is going to be a little overwhelming at first, but I want to, does this look familiar to anyone?
Yeah.
Nothing? Anything? What does this look like? We've talked about things like this before, but it looks way different. But all this really is, I've done drawings like this a couple times this whole year, where I'm drawing a capillary bed, right? It's my great artistic skill. Drawing a complicated capillary bed. Oh, that was pinched off. Yeah, no. So you got blood flowing through this capillary bed. These are red blood cells. So I've drawn pictures like this before. We talked about this last when we talked about the lymphatic system. We have a capillary bed, but capillary beds are not this simple actually. They actually have holes in them, right? They're permeable. So there's cracks along the entire capillary bed that allow fluid to leak out. And now it's not red blood cells usually. It's the plasma, it's the ions dissolved in there, the glucose dissolved in there, maybe some white blood cells squeezing through the walls. But usually the proteins remain in the capillary bed, but the watery and dissolved things come out. Now they don't just come out and stay there. Most of them are reabsorbed, about 90% are reabsorbed in a normal capillary bed. And that leaves us with one extra component of fluid that needs to be dealt with. And how did we deal with this fluid? The lymphatic system. So the lymphatic system just captured it, took it back off to the veins, right? Immune system's hanging out in there, capturing anything that's bad. But that's a normal capillary bed. So again, take a look at the nephron. and try to see some of the similarities between the two. The biggest one is that this lymphatic structure is just really well developed. So imagine this thing just evolving to totally surround the capillary bed and then just pinch it up. Take these two ends and pinch them up here, and that's all you're looking at there. This is just, the glomerulus is simply a fancy name for a modified capillary bed. It's a capillary bed. That's a glomerulus if I just pinch this and pull it up like that and surround it with a lymphatic duct. And so this is often in the beginning of textbooks described as a highly modified capillary bed. Other big difference, if we look at where this fluid is going, now if this were a lymphatic vessel, this fluid would make its way back to the veins to keep your blood pressure the same, keep your volume in your blood the same. In this case, though, we don't do that. We send it off eventually out, and this would go to the renal pyramid. So let me just lift this up so I could draw on it. So this duct here, the collecting duct, would be at our renal pyramid. And so it would be being captured by that minor calyx. Be going out that way in the kidney in the grand scheme of things. Totally out of proportion. The duct's way smaller than that. But so that's one big modification to it. It's not going back to the venous structure. It's going out into the toilet. Now, if we talk about each one of these structures, arterioles we've mentioned before. Arterioles are just the last vessels that still have smooth muscle on them that control, so this circle I'm drawing is a sphincter. It's representing a sphincter in the arteriole that leads to this normal capillary bed over here. If I constrict this or dilate this, I'm going to control blood flow through the capillary bed. Well, you also have that same function going into this capillary bed because this one basically evolved from this one. And so you have, it just has a special name. It's called the afferent arteriole. Afferent means to bring to. If you remember, that's another term that's used for neurons. Neurons coming in, sensory neurons, another name for them are afferent neurons. So this is the blood going to the glomerulus. And then you also have an efferent arteriole, which is leading away from the glomerulus. Pretty straightforward. Now, the blood flow continues on a pretty specific pathway. After you go from the efferent arteriole, this tube right here, beginning, I'd say right there, and going to right here, this is called the peritubular capillary. So this is a capillary that is most important because it's right next to this proximal convoluted tubule. Did you guys talk about the proximal convoluted tubule yet? Any functional stuff, or just like that's the proximal convoluted tubule? No functional. We'll get to the function in a second. But that is the peritubular capillary. We'll talk about why that's important in a second. I just want to name out the anatomy. We're going to go to the end here. Our last blood vessel is called the vasa recta, and it's labeled right there. And it is the loop, it is the part of this capillary bed. So this is this is one capillary here. Then it narrows to an arteriole again. So arteriole, capillary, arteriole, efferent arteriole. And this is actually another capillary bed. So these are multiple capillary beds in series. And the vasa recta is also a capillary bed. It just follows the loop of Henly. And then eventually it converges, and this would turn into venous blood flow, and it would return to your circulation at the end of this. Now we look at the renal tubules. That's a general term for the blue stuff that you see in this picture. But you're usually not going to be talking about this in that general of a term. Normally you're going to be speaking about specifically the first part of the tubule is what's called the Bowman's capsule. It's what's actually surrounding the glomerulus and capturing the filtered blood. Then there's a proximal convoluted tubule, the loop of Henly, which has a descending limb and an ascending limb because filtrate is moving in this direction. Then you've got a distal convoluted tubule, which empties into the collecting duct, which eventually empties into a minor calyx. So that's the flow of filtrate. And that's the basic anatomy that you guys are going to need to be able to pick out for this week. But let's think about also, again, bringing it back to what a normal capillary bed does. just like this capillary bed filters and reabsorbs, this capillary bed does the same thing. So early on, blood is coming in here. It's going to filter out into this Bowman's capsule, and it actually filters a lot. That is one way that it's really different. It filters way more than a normal capillary bed. That's why there's more arrows. It reabsorbs still, for the same reasons, but it doesn't reabsorb quite as much. So you get a massive filtration into the Bowman's capsule. And remember, all that's being filtered in here, you're not filtering proteins, you're not filtering red blood cells, you're just filtering out a ton of water, a lot of ions, a lot of glucose, anything that's really water soluble and small, basically. So that's what's coming out into the proximal convoluted tubule. Now, that's going to be a problem for a couple reasons. If that was the end of the story, if you just peed this away, if there's nothing else going on and that just ended up in the toilet, that'd be a problem because about 40% of the volume that comes in to this capillary bed is filtered out. So you're going to lose, if that was the end of the story, if this just went off into the toilet, then you would run out of blood very quickly because you're losing a lot of water. And you get rid of all those valuable things like ions, all those different kinds of ions and glucose, most importantly. You would just be peeing it away. So you don't do that. Instead, you send it to the proximal convoluted tubule, which is a huge tube because it's got a lot of volume in it. It's dealing with all of that fluid that you filtered. And you do two things there. You go through what's called reabsorption. So these red arrows are going back to the capillary bed from the PCT. That's specifically tubular reabsorption. So those arrows that I just drew just represent recapturing water, glucose, and ions. That's pretty much what you're recapturing. That way you don't just pee them away. So that's one of the two main functions of the proximal convoluted tubule. You're pushing out everything, basically, except for protein and red blood cells here. And then you're grabbing all the stuff you still want to hold on to so you don't waste it away. That's reabsorption. Secretion is just the opposite. It happens a little later in the convoluted tubule, but it's actually pushing things that were too big to be filtered, like they couldn't fit through the holes in the capillary bed, but you still want to get rid of them. Those will remain in the peritubular capillaries, but you can still get rid of them through secretion. And so things that do this a lot are often medications. We have the transporters that are used here will grab onto things with sort of a common shape that your body has evolved to somehow know is something you want to get rid of. And so like when you take antibiotics and it changes the color of your urine, it's not because you filtered out the antibiotics, it's because those antibiotics are actually really large, they couldn't fit out. Instead, they go to the peritubular capillary and are secreted at the proximal convoluted tubule. And so those are the two major functions. Reabsorption, really important, because you just blindly dump stuff into the PCT. You want to grab all the stuff you want, and then if you're the blood's running over here and it's got stuff you don't want in the blood, you push it through and you dump it into the proximal convoluted tubule. So ideally, by the time by the time the filtrate gets here, it's full of a bunch of stuff you don't want, and it has almost nothing that you do want. So then that filtrate is going to go through the loop of Henly. Not any significant modifications to it there. You're not really like reabsorbing or secreting anything more that you want to hold on to. Then you're going to get into the distal convoluted tubule, which has a lot of functions regarding hormones. This is where most hormones are acting on the kidney, and we're not going to talk about that this week. But ideally, it just remains flowing through here, dumps into the convoluted tubule, and then there's one last thing that can happen there that we'll get to in a second. Does that make sense, kind of big picture, how this is working? Because I think that's a really important thing. A lot of times you get lost in all these individual details, and the next class on renal physiology you take, you're probably going to have to memorize mechanisms for how this happens. But the big picture, again, it's capillary bed. You dump everything into the PCT. You grab what you want from the PCT, that's reabsorption, and then you take out a couple last second things that you want out of the blood, and that's secretion. So that's why the PCT is probably the most important part of the kidney in terms of regulation. A lot of time is spent studying how this stuff works. This is a better way to get rid of things too. If you design a pharmaceutical or some kind of drug, or you just find a drug or something like that medicinally, naturally in the environment, it'd be better if you could get rid of it like this. because this is just dumping. This is just dumping the trash, basically. If it can't be secreted this way, it will remain in your blood. And then the only thing left to deal with it is the liver. And the liver can do some cool stuff, right? It can convert glucose into fats and fats into glucose. But a lot of times when it comes to metabolism of drugs, it tends to harm your liver. That's why you're usually not supposed to like take drugs for the rest of your life. If you have to, you have to. But you shouldn't take mega doses of aspirin every single day, because that aspirin actually isn't secreted. It has to be metabolized by the liver, and it can hurt your liver in the long term. So this is a good thing. They aim to make drugs that are secreted, because then you just pee them away. Great. Don't even have to use the liver. This is also how the compound in asparagus gets into your urine so quickly. So if you eat asparagus tonight, start a stopwatch when you put it in your mouth, and it should be in your urine in about 12 minutes. It's that quick. And if you think about digestion, it kind of ties together the whole last couple of weeks that we've been talking about, right? It's going to get absorbed mostly in the small intestine. That compound's going to go through the liver real quickly into the circulation. Has a 1 in 5 chance of ending up in the kidneys on first pass. You're going to immediately secrete it. It's going to stay in here, and it's going to end up in your toilet. So any questions about the proximal convoluted tubule? Because the last thing we're going to talk about is the loop of Henle and how the collecting duct uses the loop of Henle. All right, so the loop of Henle, the main function of the loop of Henle is to create a concentration gradient. And it's the most important concentration gradient in your body. So remember from our previous picture, the loop of Henle is in the medulla of the kidney, whereas everything else, except for this part of the collecting duct, everything from about here up is in the cortex. So what happens is that this vasa recta actually pumps out salts into the medulla. Sorry, not the vasa recta. The loop of Henle pumps out salts into the medulla and creates a very salty environment from about here down. So that red line, that's basically representing the medulla. So everything below this line becomes extremely salty, about 3 times saltier than your normal body. We're kind of salty organisms, right? But we're not that salty. We're not like ocean salty. This stuff down here in the medulla is almost as salty as the ocean. The ocean is about 1,000 what are called milliosmoles, which is a way to measure salt concentration. This is very close to 1,000, but not quite 1,000. So we'll just say 999, basically from here down. That just represents the salt concentration. The higher the number, the saltier it is. And I'll just put a comparative number there. This is about 300. That's your normal concentration of salt in every cell in your body. So you create this gradient here. And so everything down here is much saltier than everything up here. And because they're anatomically distinct region, one's the cortex, one's the medulla, it doesn't equalize across here very quickly. So the salts are being pumped out by the loop of Henle, creating this salty environment. But you probably ask yourself, Greg, why the hell do we do this? Why would we create a salty environment? Isn't that going to cause water to rush out of cells and cause a lot of osmosis issues? Well, yeah, it actually does, but we utilize one of the consequences of osmosis to our advantage. So here's how we do this. So we've already talked about how this fluid in the blue tube here should be full of things we don't want, but there's still going to be a little water in it, right? Everything here still contains water. Well, what happens is the fluid that makes its way over here actually is quite low, has a very low salt concentration. So our normal cells are 300, but the fluid in here is about 90 milliosmoles at that point in the tube, at the top of the tube, right after it's come out of a DCT. So it's actually really dilute. Now we've seen 90 milliosmol urine before. That's urine that has almost no color in it. So all urine has a little color in it, but you know, when you pee it, it's just like water. It's like you didn't even do anything. It's totally water, almost totally water. That's that concentration right there. And if you are properly hydrated, that's the end of the story. This just continues down the collecting duct and takes that long pathway into the toilet. But if you are dehydrated or have a low blood pressure, so if you need, both of those things are a need for more water. Your low blood pressure, your body says, well, throw water in your arteries and veins to boost your blood pressure. And if you are too salty, and your hypothalamus regulates that, it'll send a message down to basically your entire body, but it'll get to the kidney to say, hey, we need to hold on to more water. So as this 90 comes down, this very, very dilute, basically fresh water moves into this highly concentrated region, it will actually, at the same time, you will put these little holes in the collecting duct. These are called aquaporins. And those holes allow only water to move. That's why they're called aquaporins, aqua meaning water. So as this dilute fluid moves down into this very salty fluid, you have like a classic example of osmosis. You have a semi-permeable membrane with a large salt concentration out here and a very small one here. What's going to happen to the water? Which direction is water always going to go? Towards the salt. It's going to flow out. This is dilute water. This is highly concentrated water. Now you might be thinking, wait, why don't the ions move that way? They would, but they can't. They can't cross that membrane. It's only permeable to water, so the only thing left to happen is this. And so all the water that moves through the aquaporins is recovered. This is your body holding on to water that would have otherwise ended up in the toilet. And so this helps you preserve water, put it back into the vasa recta. That's exactly where it goes. It hydrates the vasa recta and returns to the circulation. So you're going to learn about a hormone called ADH, antidiuretic hormone. Diuresis is losing a lot of water through the urine. So the hormone that's called antidiuretic hormone just does the opposite of that. When you release ADH, you insert aquaporins, and you use this expensive, it took a lot of work to build this gradient, but you use the gradient that the vasa recta and the loop of Henly have created together. Yeah.
So ADH actually creates the aquaporins or just opens them?
It inserts them is always how it's said.
Oh.
Create, I'm guessing they're resting.
They weren't there before.
They were not there before. This was a solid, the doors are closed. And if that happens, you're going to pee clear fluid because you haven't recovered any water out of it. When you have even a little bit of dehydration, you're going to insert, you're going to open up or insert, it's always referred to as inserting though. Inserting aquaporins, and that's going to allow water to flow into this region and into the vasa recta, and you're going to hold on to water. So that's the value of the loop of Henly. Yeah.
So if you're taking diuretics, it does the exact opposite? Would it open
So
up or
for diuretics can
close
work
them?
in a lot of ways. Let's use an example of alcohol. Alcohol dehydrates you. It can be quite dangerous if you drink a lot of it without drinking water. You can get dangerous in that it makes you really, really dehydrated. Alcohol inhibits ADH release. And so ADH is a hormone that circulates through your whole body, and that's what it does in your kidneys. If you have alcohol, it inhibits the release of ADH, which means even when you're dehydrated, you won't put the aquaporins in, so you'll pee away the water that you actually need. And then you'll also eat salty food at the same time, which will require more salt to dilute out, and you won't do that either, so it's a pretty nasty little feedback. There are other drugs that will inhibit ADH. The diuretics that are used clinically tend to target, if you ever hear the term loop diuretic, it targets the loop of Henly. It actually stops the loop of Henly from creating the gradient in the first place. So even when your body releases ADH, nothing's going to happen. Well, something might happen. So like anyone who's on diuretics, this number changes. Instead of 999, it's more like 800 based on the dosage. But it's about 800, which means you're still going to recover water, but not as much. And it'll bring your blood pressure down. That's the idea. Lasix, which is what they'll use in an inpatient setting usually when someone has congestive heart failure. That's the most severe of the loop diuretics. It essentially destroys this, puts it at like 310 or something. Kind of dangerous. But this is where the cost benefit is very different, because if you were to allow these individuals to keep this fluid on their lungs, they would probably die. And so Lasix, you're just going to be drinking, just chugging it down. And I do it like this because it's literally like a gallon jug that you're usually chugging. And what that's going to do is basically shut down the loop of Henly, prevent you from retaining water, so you constantly lose water. And that's to get the fluid off of people's lungs usually. And so it's a dangerous thing. It damages the kidneys permanently. a lot of these things do, but that's only in extreme situations. Normal diuretics do have side effects, but not that significant.
So what happens when parents have kids who have bedwetting problems
Mhm.
and they get medication, so essentially they're shutting off the kidneys from producing?
I'm not familiar with the drugs that they use for that.
Oh.
I mean, bedwetting is more of a psychological and your body's inability to shut down the external urethral sphincter during sleep. Because sleep is a crazy game, right? So I got this Fitbit thing, and it tells me like when I'm paralyzed and when I'm not at night, which is kind of cool, right? Because there's certain phases of sleep where you're paralyzed. Well, that's a problem in kids because the paralysis can allow them to release urine everywhere. Very common thing, but I don't know what drugs they would do. It seems kind of unnecessary to mess with the kidney if it's just pee in the bed. I mean, it might suck, but it's not going to kill anyone. But I don't know, I'm not sure what they use. They may not even use something that targets the kidney. It could be something that affects their sleep patterns in another way. I'm not really sure. You should look into it though. Because yeah, normally you run into these clinically just with blood pressure management. Diuretics are the first line of chemical defense against blood pressure. They work in a couple different ways, but they inhibit your ability to recover water. So that's kind of the main thing. So hopefully the big takeaway message is that you got to have the collecting ducts and the loop of Henle running if you want to recover water. If anything affects one of those two things working, either you don't release ADH, that's diabetes insipidus is an example of something where you can't release ADH, so the last thing I'll say before we stop talking about the nephron is this vasa recta, remember it's a capillary bed. It's the longest capillary bed in your entire body. It's inches long, like that big. And that's hundreds of times longer than most capillary beds. So there's often a connection. So type 2 diabetes results in extremely high blood glucose levels in certain situations. Type 1 diabetes can as well, but it's a little bit easier to manage. So type 2 diabetes is the more fast-growing one in the United States and worldwide, actually, now. But the reason it's connected to renal failure is because high blood glucose levels make the red blood cells much larger. They damage the red blood cells temporarily, but because your blood sugar is usually permanently somewhat higher than it should be, the red blood cells actually start to bounce off of the walls of the vasa recta. They can't move through. It's kind of like a traffic jam. And so what ends up happening is the flow of red blood cells through the vasa recta is not enough to maintain the life of the cells in the loop of Henle. These are very energetically demanding cells. They're creating a concentration gradient, which is very hard to do. You have to pay a lot of ATP for that, which means you need adequate blood flow to the entire kidney. But if you have anything that gets in the way of the blood flow, the first cells that are going to die are right here. And if you kill the cells in the loop of Henle, you can't make as good of a gradient. If you can't make as good of a gradient, you cannot retain as much water. And so this is where the symptom of lots of urination comes from in diabetes, is that even if ADH is working, even if everything else is working, if you've killed off some of your loops of Henle, you will build a smaller gradient. So instead of maybe 999 in a normal person, you're going to get down to maybe 700. So all of a sudden you're going to need to drink more water. That's where thirst is also a common complaint, constant thirst, constant urination, because you cannot retain water because you're killing off your nephrons. And so that's kind of the connection between those two things. And everyone's kidneys will eventually poop out on them. You're constantly, so to give you a context of how many of these things you have about 1 million in each kidney. And trauma is a real good way to kill off nephrons. High blood glucose is a real good way to kill off nephrons. And high blood pressure is another good way to kill off nephrons. And so those three things, if you combine them together, are something that can predispose you to a little bit earlier renal failure. But really, by the time you're like 100, your renal system's going to poop out on you anyway. But luckily, we have dialysis to deal with that. It's not a very sexy option, but it works. And in 2 weeks, we'll talk about how dialysis works, why it's going to be an increasingly prevalent thing in our society, and why you guys should probably shadow in a dialysis clinic and start to learn about that, because it's growing rapidly in our society, and it's good job opportunities, and a lot of our patients will be on dialysis. You might as well kind of learn how it works.
So you're saying that old age or getting older, just we're naturally going to
Constant decline, kind of like vital capacity. But the tricky part in terms of diagnosing at vital capacity, I can know. I'll test it this year and next year, do it, and I would easily be able to tell that my kidneys are failing, or at least going downhill, not failing. That's too significant, just degrading. Kidneys, you can do a blood draw and a normal blood panel. It'll tell you your creatinine levels, which are a rough estimate of how well your kidneys are working. However, not that many people get their blood drawn and tested. And so a lot of people have kidneys that have degraded significantly, maybe between the age of 20 and 40, but because they don't get blood tests done, they don't know. And so the only thing that tells them is they get much more significant symptoms. And that doesn't happen